Hey everyone, welcome back to my channel. Today I'm going to talk about DNA replication part 2 of elongation, and I would please request you to watch my previous video on DNA replication part 1 of initiation before watching this lesson. So DNA replication is initiated by formation of this origin of replication, and this site of origin of replication is enriched in adenine and thymine, and the pre-replication protein complex which binds to this origin of replication and creates this replication bubble, and the ends of this replication bubble, they are called replication fork, and that's where helicase enzyme comes in and start unwinding these two DNA strands. And while doing that, it creates this torsional strain between these two DNA strands that can result into supercoiling of these two DNA strands, and that is prevented by topoisomerase enzyme. And this separated two DNA strands, they have a high affinity for each other, so they are very vulnerable to reconnect or rejoin, and that can be prevented by this single stranded binding protein. So this SSBP protein binds to this single stranded DNA and keep them apart, as well as it also protect this two strands from nucleases enzyme, which actually degrade this single stranded DNA. So that is the initiation process of DNA replication. So today in this lesson, let's talk about how these new strands, or how these two daughter strands are being synthesized. So there is one very important enzyme which play a role in synthesizing new DNA chain. This enzyme is called DNA polymerase. So now for DNA polymerase to catalyze the synthesis of new DNA strand, it must be provided with three things. First, a template DNA strand. So suppose we already learned this process, how these DNA strands are separated. So this is our two parent DNA strands, and this two parent DNA strand will be used as a template to synthesize new DNA chain. Second, it also requires 4-deoxynucleoside triphosphate. Now, deoxynucleoside because this is used to synthesize DNA, so that's why it's called deoxynucleosides. So now here we have a nucleoside. So if you remember from my previous video, nucleosides are is the connection of sugar molecule to nitrogenous base. So this is our nucleoside. Now triphosphate, because this nucleosides has 3-phosphate group which is attached to the 5' position of the sugar molecule. So that's why it is called deoxynucleoside. So deoxynucleoside triphosphate, that is DNTP. Now I have mentioned here 4 because there are 4 different nitrogenous bases, if you know, for DNA, which is adenine, thymine, guanine, and cytosine. So depending on the nitrogenous base, there are 4 different types of deoxynucleoside triphosphate. So that will be adenosine triphosphate, cytosine triphosphate, thymidine, and guanosine triphosphate. So there are 4. So we need these 4 deoxynucleoside triphosphate in order for DNA polymerase to initiate synthesizing the new DNA chain. However, the question is, why does DNA polymerase require 3-phosphate group? Why can't it manage it with only 1 phosphate group? Now the reason behind is synthesizing new DNA strand is a very energy consuming process. So what nucleoside does is they actually brings their own energy in the form of 3-phosphate along with them, and those phosphate groups will be utilized to synthesize this new strand, and you will see this shortly how this phosphate group is being used to synthesize new strand. Now last thing DNA polymerase also need is RNA primer. Now, this is weird, right? Why do we need RNA primer to synthesize DNA? Now, RNA primer is nothing but it's just a short strand of single-stranded ribonucleotides which are synthesized by enzyme primase. Now the question is, why do we need this? So it turns out that DNA polymerase cannot just start adding this nucleoside triphosphate to synthesize new DNA strand. It actually need this free 3-hydroxyl group which is attached to the 3' position of the sugar molecule. If it does not have this, it cannot start synthesizing this new strand of DNA. And this 3-hydroxyl group is important to connect the second nucleotide. So if you remember that the phosphodiester bond is formed between the phosphate group of one nucleotide and to the hydroxyl group of the second nucleotide, which is located on the 3' position. So therefore, if DNA polymerase does not have this free 3-hydroxyl group, it cannot actually start synthesizing the new strand. So basically, the main function of RNA primer is to provide this 3-hydroxyl group as a starting material, which DNA polymerase then can use to start building the new DNA strand. So basically, the main function of RNA primer is to provide this 3-hydroxyl group as a starting material, which DNA polymerase then can use to start building the new DNA strand. So let's see how this is going to happen. So suppose we have this two parental DNA strand which are separated, and RNA primer comes in. It actually brings this ribonucleotides and these ribonucleotides which are complementary to the parental DNA strands. So I just want to mention or highlight this here, that RNA does not have thymine. Instead of thymine, it has uracil. So here adenine actually pairs up with uracil. So just want to bring that up, that you don't really get confused that why is adenine is pairing with uracil. So now once RNA primer comes in and brings the ribonucleotide and attaches brings the complementary ribonucleotides, what will happen that it actually now provides the starting material. And what is the starting material I talked about? That is the 3' free, that is the free hydroxyl group which is attached to the 3' position of the sugar molecule. So now once DNA polymerase has this starting material, it can now start elongating this chain from here onwards and start synthesizing the new DNA strand. So now another important thing I want to mention, that as I talked before, that DNA new strand can only synthesize from 5' to 3' end. So here you can see that we have a 3' position, so the new strand is being synthesized in this direction from 5 to 3. So now let's see how DNA polymerase elongate the new strand from here onwards. So DNA polymerase 3 in prokaryotes, which is responsible for elongating this DNA chain from here onwards. So let's see, DNA polymerase brings in this nucleoside triphosphate, and what it will do is it will actually cleave this 2-phosphate group, which is called pyrophosphate, and subsequently this pyrophosphate is converted into 2-orthophosphate, and this is actually the powerhouse of the energy. When pyrophosphate is converted into 2-orthophosphate, it will release this energy, and this energy will be then utilized to form the phosphodiester bond between the phosphate group of this nucleoside and the hydroxyl group on this sugar molecule. So therefore you now know that why is this 3' hydroxyl group is important for DNA polymerase. And these two nitrogenous bases are connected to each other via a non-covalent hydrogen bonds. So let's see it again. DNA polymerase comes in with the nucleoside triphosphate, releases pyrophosphate, and which again releases the energy which is being utilized to build this phosphodiester bond. Let's see it again. Polymerase cleaves this pyrophosphate, releases energy, and then build this phosphodiester bond. And likewise, let's just complete this side of the strand. So now you can see that we have a new DNA strand which is synthesized in 5' to 3' direction. So I also want to mention one thing, which is the importance of 3-hydroxyl group. So there are this class of drugs which are called nucleoside analogs. These are acyclovir, cytarabine, and azithromycin. So these are antiviral medication which is used to treat herpes simplex virus, chickenpox, as well as shingles. So what they do is they actually inhibit the DNA polymerase enzyme. The way it does is this nucleoside analog they actually lack this 3-hydroxyl group here. So if you don't have this 3-hydroxyl group, DNA polymerase cannot elongate the new DNA strand. So therefore it competitively inhibit this DNA polymerase enzyme, and that way it arrests the viral DNA replication. So now let's talk about how the new chain, how can we synthesize the new DNA strand on this side, because it is not as easy as this side of the DNA strand, because if you see here we have this 5' phosphate group, and we can only, and then the opposite to that will be 3' hydroxyl group. Unfortunately we cannot synthesize from 3 to 5, and it must synthesize from 5 to 3. So let's see how that is going to happen. So just imagine that Helicase has only unwinded this much portion of DNA strand. So as soon as Helicase start unwinding the strand, DNA polymerase will start synthesizing the new DNA strand from 5' to 3' end. So just as we saw before, the starting material will be provided by the RNA primer. So RNA primer comes in, bring its own ribonucleotides, and then from here DNA polymerase will start extending the strand. So here it completes this DNA strands. Now let's say Helicase unwinded some more DNA strand. So what it will do is, again it will synthesize another small piece of DNA strand. Let's say Helicase unwinded some more DNA strand. So again there is new strand which is again synthesized in small pieces. So on this side of the strand, it actually synthesized in 5 to 3' direction. However, it's synthesized in small pieces, if you can see here. Why? Because if you see here, these strands are not connected to each other. There is no phosphodiester bond here. That means there are these pieces of strands, or there are fragments of these strands which are actually called Okazaki fragments. So it is opposite to this. Then this strand synthesized in a discontinuous manner. So that's why it is called as discontinuous or lagging strand. And this strand which continuously synthesized from 5' to 3' end, therefore it is called as continuous or leading strand. And the another thing to know is there is only one single RNA primer which is required to synthesize leading strand, whereas there are multiple RNA primer which is required to build this lagging strand. Now another thing we need to know that DNA polymerase also has proofreading function. So proofreading is very important when new strands are synthesized. So what DNA polymerase will do is it will actually proofread the nucleotide sequence of these new strands to make sure that the base pairing is proper. If there is any mistake or if there is any error in the base pairing, that can have very very detrimental consequences. It can lead to mutations and perhaps it can be even lethal. So it is important to make sure that this DNA replication takes place with as few error as possible, and that is done by proofreading these nucleotide strands, and that is performed by DNA polymerase enzyme. So proofreading is performed by DNA polymerase 1 and DNA polymerase 3 in prokaryotic cells. So it performs this exonuclease activity. Exonuclease means it actually removes one nucleotide at a time and replaces with another nucleotide. So if it finds any error in the base pairing, it will actually remove the nucleotide and then replaces with the correct nucleotide. And I have mentioned here 3 prime to 5 prime because it moves in 3 prime to 5 prime direction to proofread the nucleotide sequence. So let's say here we have DNA polymerase 1 or 3 enzyme. It will move in this 3 prime to 5 prime direction. Now it stopped here because it encountered an error. So if you can see here that guanosine does not really pair with thymine, so which is wrong. So what it is going to do is it is going to remove this nucleotide and replaces with the correct complementary base pair, that is cytosine. So this is how DNA polymerase proofreads the proofreads the nucleotide sequence and correct the errors which is being done during the synthesis of the new chain. Now another thing is excision of RNA primer. So RNA primer, we saw that they already serve their purpose. They already provided the starting material for DNA polymerase to extend the new strand, and that starting material, if you remember, is that 3 prime hydroxyl group. So they already provided this starting material. Now we don't need them anymore. They must be removed because this is DNA strand, and we cannot really have these ribonucleotides in DNA strand. So they must be removed, and that is called excision of RNA primer, and that is performed by DNA polymerase 1 in prokaryotic cell. So they also perform exonuclease activity in 5 prime to 3 prime direction. So exonuclease, again, it is going to remove this ribonucleotide and replaces with deoxynucleotide. So suppose we have this we have this DNA polymerase enzyme here. It locates this RNA primer, remove this ribonucleotide and replaces with deoxynucleotide. Now you must be wondering that, I just took out the cytosine and replaced it with cytosine, but there is still a difference. The sugar molecule are different. In ribonucleotide, the sugar molecule is ribose sugar, and in deoxynucleotide, the sugar molecule is deoxyribose sugar. So that's why I'm still replacing it. Then it moves further and locate another RNA primer, remove this ribonucleotide, replaces with deoxynucleotide. Likewise, it will also remove this ribonucleotide. This uracil is replaced by thymine, and it will also do the same on this side of the strand and replaces with deoxynucleotide. So now while it is moving from 5 prime to 3 prime direction, it is removing the RNA primer, replacing it with the deoxynucleotide. So this replacing of ribonucleotide with deoxynucleotide, this again needs to be proofread. It cannot just It cannot just think that, oh, I replaced this deoxynucleotide and now new DNA strand is completed. It must proofread whenever it make any changes. It must proofread to make sure that the nucleotide sequence is correct. So what it is going to do is it is going to perform another exonuclease activity from 3' end to 5' end to proofread the nucleotide sequence to make sure that correct base pairing, to make sure that there is a correct base pair between the daughter strand and the parental strand. So that means DNA polymerase has three important things to do. First, it removes the RNA primer, and when it by removing the ribonucleotides, and when it removes that, it synthesizes the new strand by replacing this ribonucleotide with deoxynucleotide, and when it does that, it eventually has to proofread the new DNA strand to make sure that all the base pairings are correct. So in short, DNA polymerase perform the exonuclease activity from 5' to 3' direction to remove RNA primer, and it will and then it will go back in 3' to 5' direction to proofread the nucleotide sequence to make sure that all the complementary base pairs are correct. Now lastly, we still have this Okazaki fragments, which are still the fragments, and these strands are still not connected. If they are not connected, the formation of the new strand is still not completed. So there is one last enzyme which comes and play an important role, that is called DNA ligase. So DNA ligase comes in, it provides an energy in the form of ATP to build this phosphodiester bond between these two strands, which are fragments. So phosphodiester bond is built between the phosphate group and the sugar molecule of phosphate group of one strand and the sugar molecule of the other strand. So that way it seals this Okazaki fragment to synthesize, to complete the synthesis of lagging strand. So lastly, let's just have a quick recap. So we have this replication fork here. Helicase enzyme comes in, which separate these two strands, and while doing that, it can also cause DNA supercoiling, which is inhibited by topoisomerase enzyme, and these two strands have high affinity for each other to reseal or reconnecting. So the single-stranded binding protein which comes in, which binds with the single-stranded DNA to prevent the reannealing or reconnecting. And then the new strand, the starting material for the to synthesize the new strand is provided by RNA primer, and DNA polymerase comes in, which actually start in elongating the new DNA strand from here onwards. So it builds this continuous or leading strand on this side, and on this side it builds the discontinuous and the lagging strand. Now the lagging strand has these fragments of DNA strands which are called Okazaki fragment, and eventually these DNA fragments are sealed with the help of DNA ligase enzyme to finally complete the synthesis of lagging strand. So this lesson and my previous lesson complete the DNA replication process of initiation and elongation. However, we still need to know how DNA replication is being terminated. So I'll be making a separate video on DNA replication part 3 of termination. So thank you so much for watching this video, guys, and if you feel you learned something from this video, please like and share the video and subscribe the channel. Thanks so much and have a great day.